Insect Newick review of Jin et al. 2022 phylogeny of Cerambycids. These are longhorn beetles, plant-eating beetles, and highly diverse. Download, and we get a bunch of files. Now, the first tree in their paper is maximum likelihood full degenerate nucleotide partitioned by codon position. So I'm guessing that will be this one. So I've copied this into the working directory and changed the name to this, and this is what we've got to work with. Cat file, and we get this in node support, branch length, and terminal IDs use codes. Majority start in ANIC, then 3 numbers. So we need to figure out what species these are. So in the only closest I can find in this supplement is this table. So they do have these codes ANIC for the lab ID, then an extra number which seems not applicable, and they have the species names for each of these. Now they don't have exactly the same look. They've got a underscore and for some reason the word speeds after. Don't know why that's. So they haven't got the terminal IDs, but they do have the codes. Ideally, these would exactly match. So we'll try and see if we can match with just these codes. So first thing is to turn this into a mapping file. So just copy these codes into a text file. These are tab separated. So we start with some standard code, and then this will need adapting to this. So we open first the mapping file, tab separated. Let's just take a quick look at this. I think I would just remove these dots from the SP. So it's reading the codes and the species. Okay, it's not exactly. Okay, I'm going to assume that is the genus. I think this means NR. That's referring to the genus, not the species. But open tree of life, assume when it's NR, it will just assume the correctness. And I think I'll do the same. Otherwise, this looks all quite neat and tidy. So let's go to the next step. So we've just read for each code, that's the key of the hash. Hash is called species of code, and then put the species name in there. Next, go read the tree, and then here's the key part here. Loop through each code and try and find it in the Newick string. So these single quotes, this is specific for Open Tree of Life format. So I'm going to remove this because I'm not using that format. Now, so the main difficulty is the code. This table doesn't give it's not specifically given the terminal IDs. It's giving codes which are within the terminal IDs. So quickly looking at this. So some of them. Okay, maybe a simple way to maybe just remove this. Seems to be mostly. I don't know, it's quite an odd thing to do, just to write underscore speeds after the majority of the. But maybe just remove this string here, and maybe that will fix it. So here's where the tree is read. tree is read into variable called tree. Put a G so remove all instances of underscore speeds. And then go through all the codes. It's looking for open bracket or comma, then a code, then this probably should be, okay, shouldn't be, this should be a closed bracket, then a comma, then a colon denoting a branch length. Then swap the code. Okay, see if this works. And it looks pretty good. I'm going to remove these node support. This looks pretty good. So 35 species ambiguous terminals, and all of them are genus unique. So that's perfectly good. Well, that's quite sensible, and I would keep all these. So here's the processed version with the standard, with the preferred format for the terminals, and we just need to check it's oriented as per figure. Okay, good job with checklist. This seems to be unrooted. So we have, so it's the search for these two things to denote the main group, and they're stuck right in the middle. So I'm going to click node to root, reroute, and let's try here. Two major clades which look like these two. Just find some taxa which define these. Okay, these are two. Here's a taxon at the base of this clade and one here. Okay, so this seems to be how it's oriented as far as I can see. So now we save tree in this orientation. And here's our process tree. Process tree, and ready to go. Job done.